Okay, hi everyone. So today I will be talking to you about putting things the right way around and the identification of upside down jellyfish in Lake Macquarie, which is a temperate coastal lake on the east coast of Australia. So jellyfish blooms are renowned for their bloom and bust nature. What this means is their populations rapidly increase in a bloom, followed by a population crash referred to as a bust. Now, it's believed that jellyfish blooms are increasing on a global scale due to increased favourable conditions as a result of human modifications to coastal environments. So this includes increased man-made structures, climate change causing increased temperature, eutrophication, and changes to salinity, as well as fisheries depleting predators and competitors. So jellyfish are also expanding their range through both natural means and anthropogenic means, such as shipping traffic, ballast water, or release through the aquarium trade. So my focal species is the upside down jellyfish, which is the genus Cassiopea. Now, these are kind of like the lazy jellyfish of the sea. They spend most of their life upside down with their bell resting on the sediment and then their oral arms extending above them. They do this because they have photosynthetic algae called zooxanthellae in their tissues, which provides the jellyfish with up to 90% of their nutritional needs, with the other 10% coming from filter feeding. And as you can see from this picture down here, they can swim when they're so required to do so. So upside down jellyfish typically occur in tropical to subtropical regions. So they're typically in very sheltered, protected habitats, such as mangrove forests, seagrass beds, or coral reefs. They're globally invasive with a number of range expansion records. This is happening all over the world, including Hawaii, Brazil, Vietnam, and Australia. And they're considered to be successful invaders because they have high reproductive rates and they're able to reproduce both sexually and asexually. So this is a photo of one of their invasion events in Brazil. And as you can see here, they just blanket this beach. They just occur in such high densities. In one locality overseas, they've been recorded up to 168 individuals per meter squared. Now these jellyfish can be the size of a dinner plate, so you can just imagine the ecological impact that this can have. So they are considered to be ecosystem engineers. This is because they can have direct impacts such as predation or indirect impacts such as competition. Additionally, these jellyfish release their stinging cells into the water column, and this can cause local fish to leave the area. So upside down jellyfish within Australia typically have a more northern tropical distribution. There is this one population in South Australia that's been there for over 20 years and occurs by a power plant outlet, so the water is a lot warmer there. But my main interest is these three red dots here. That's because they occur in temperate New South Wales. So the jellyfish were first recorded in Lake Illawarra in 2009 and then Wallace Lake in 2014. And then a study done by my supervisors at the Australian Museum discovered through morphology that there were two species occurring. One being Cassiopea marimetans in Wallace Lake and the other Cassiopea nidrosia in Lake Illawarra. But then in 2017, a member of the public recorded that the jellyfish were occurring in Lake Macquarie. So this raised the question of which species was occurring, where had it come from, and what was the population dynamics of this jellyfish like in a temperate environment. So this kind of created the foundations for my PhD. And today I'll be talking to you about the process I went through to identify the species that was occurring. So this hasn't been an easy task. It turns out that Cassiopea are considered to be a cryptic genus, which means they're really hard to distinguish based on morphological characters alone. There are worldwide 9 species that are currently recognized. 4 of those are in Australia. And in order to identify the species, I needed to combine genetics with morphology. So for the genetics, I collected tissue samples from 2 parts of the jellyfish, the first being the tip of the oral arm and the second being the gonads. We used 2 parts of the tissue because we found that it was really hard to extract DNA from the jellyfish. We think this is because they're quite mucousy. So this is a neat little video of me extracting the gonads from the jellyfish. You can see they kind of just slide out of the subgenital pit, and then we put it in 95% ethanol. And we compared the sequences through the cytochrome oxidase subunit 1 gene. And these were the results within Australia. So the Lake Macquarie specimens came out as being the same species as in Wallace Lake and Pelican Waters, which is up here in Queensland, so the more northern red dot. This is interesting because the Pelican Waters specimens, that's the type locality for Cassiopea marimetans. So that confirms what species was occurring. In green, we were able to show that in Lake Illawarra, we had a different species, which was also occurring in Coomba Creek in Queensland and up in the Northern Territory. Additionally, we have a separate species occurring in South Australia. So from here, we wanted to then see how these sequences sit worldwide. So that created this big, complicated looking tree, and I'm just going to focus on this section here. That's because this is where the Lake Macquarie specimens came out. So interestingly, the Lake Macquarie specimens came out as the same species occurring in Hawaii, Brazil, Panama, and the Florida Keys. Also really interestingly is the South Australia specimens came out as the same species as in Brazil, Mexico, French Polynesia, Egypt, Israel, Bermuda, Hawaii, and the Florida Keys. So you can see how dispersed just one species of this jellyfish can be. So from this, we're actually able to make a couple of conclusions here. Firstly, the Lake Macquarie specimens were in fact Cassiopea zamacana. Secondly, the Pelican Water specimens, which as I mentioned before, is the type locality of Cassiopea marimetans, came out as Cassiopea zamacana. So Cassiopea marimetans is in fact a junior synonym of zamacana. Another thing I'd really like to point out here is how closely related these two species are. So it seems like Cassiopea zamacana is a recent divergence from Cassiopea andromeda. So from here, I also just want to point out this green box down here. So this is where the Lake Illawarra specimens came out. They came out pretty closely related to some sequences from Papua New Guinea, Hawaii, and Japan. And for the rest of this talk, I'll just be referring to them as Cassiopea sp because no name has yet been assigned. So from here, I wanted to look at the morphological differences between the species and see if there are any distinguishing characters. As I mentioned before, they are a cryptic genus. So there's a few features I looked at here. Firstly is the lappet shape. So these are the little grooves that are occurring around the bell margin of the jellyfish. I then looked at the oral arm length and the branching pattern. So this is an oral arm here, and then the oral arm has lots of branches, little lateral branches that come off it, kind of like a tree. And then I also looked at the large appendages. So the large appendages are these leaf-like structures here, and these have two main purposes. The first one is extra surface area for the zooxanthellae, and the second one is they kind of contain little nest-like structures called Cassiopeidae, which contain the stinging cells of the jellyfish. So once I took these measurements, I compared them with a Permanova, NMDS, and SIMPA. So we actually got some really cool results. We got a significant difference for the Permanova between species, and we got some really nice clustering. So zamacana is in blue compared to Cassiopea sp in red, and then species 2, which is from Papua New Guinea, in green. Something I'd like to point out is these blue circles here, that one there is the holotype of Cassiopea marimetans. So it kind of just confirms with the nesting that morphologically marimetans was the same as zamacana. There are a few different features that the SIMPA pointed out that were the main factors driving the differences between species. This included the number of central appendages, where the largest appendage was located, and the length of the large appendage on the arm. So those are those leaf-like structures I mentioned before. Additionally, the location of the fork in the oral arm and the oral arm branching pattern and the lappet shape. So I'll just show you a couple of these. So you can see the lappet shape is for Lake Macquarie very rounded with no real grooves between them. This is the same for Pelican Waters, compared to Coomba Creek, which is Cassiopea sp, where the lappets are much more flat with notches occurring in between them. Additionally, the oral arm branching pattern in Lake Macquarie, the oral arms were alternating, and then in the fork, one side of the fork was much stronger than the other. This was the same for Pelican Waters, where again, they were alternating with one fork much stronger than the other, compared to Coomba Creek, where you had a combination of alternating and pinnate, meaning in line with each other. And additionally, the fork was equal in strength. Finally, those large appendages, these ones occurred in the fork of the arm for both Lake Macquarie and Walking Waters, compared to Coomba Creek, where there was no large appendage. So from here, we're able to conclude that Cassiopea zamacana is occurring in Lake Macquarie. Cassiopea meramitans is a junior synonym of zamacana. There appears to be 2 species moving down the east coast of Australia, so it's really important to be able to define those species so we can track their spread. We also think there was most likely 1 invasion event. We believe this because I was checking the sex of the jellyfish as I sampled their gonads, and we actually only found females. So we think there was only 1 invasion event, and then the jellyfish have been asexually reproducing. From here, I'm going to combine this data with 3 years worth of field data to understand the population dynamics of the jellyfish in a temperate environment. We also want to understand what's driving their invasion, as well as controlling their invasion front. And finally, we'd like to assign a name to the Coomba Creek and Lake Illawarra species, but that's probably post-PhD. Thank you for listening to my talk.